Welcome back, everybody. Just before we get started today on translation, I want everybody to know it's going to be a little sloppy. The pen that I use to write my lectures on my Surface Pro that I have is broken. So I am using my finger, which is even sloppier than usual. And another thing is that my internet has not been working at home. So I've been kind of working in minimal amounts the past 2 days, trying to get a few things done, but hopefully this will finish up the section on gene to protein, which was chapter 17, pretty well with this lecture. And so today's lecture is going to talk about translation. And as I said, sorry about the horrible handwriting. But translation is going to be where we take that messenger RNA, the mRNA, that we had from our last series on transcription. If you haven't watched it yet, go watch it. And we're going to take it, and it's going to be read by the ribosome, and we're going to produce an amino acid chain from it. Okay. And so that amino acid chain that's going to be made will be eventually our working protein once it's properly folded. Hopefully you remember from Unit 1, we talked about proteins and how proteins have a primary, secondary, tertiary, and quaternary structure. And they have to fold and fold and fold. And we actually made those models using pipe cleaners. And they don't work until they are properly folded. So not only do we have to make the protein correctly in the correct amino acid sequence, which is what is going to happen in translation. Translation is the process of making the amino acid sequence. Then that amino acid sequence then has to fold, okay, to make it a functional protein. So at the end of this process, we don't technically have a functional protein. It has to fold in order to make it functional. And so the first thing I want to talk to you about is we're going to talk about a couple different types of RNA today. mRNA was going to be what came from transcription, okay, and you can see it over in this diagram in red. Another type of RNA that's very important here is called tRNA. I'm going to go ahead and circle it right over here as small as I can. I'm sorry with my finger. The tRNA that is over here, that's what it looks like. And we're going to talk about the differences that make up tRNA. Now, we draw a very vague picture of tRNA. As you can see there, it looks like this little tube that has amino acids attached to the top, and then this 3 bases at the bottom there you can kind of see in the picture. But what it really looks like, it looks like a lot of bases, okay, just because it is an RNA. It's called transfer RNA. What it actually looks like is it has this little bulb on this side. It comes down here to another little bulb. Same thing on this end. And then up at the top, it's going to attach to an amino acid chain, some type of amino acid sequence at the top. Now at the bottom down here, there's a region on this tRNA that is called an anticodon. Now I'm assuming you're familiar with the term codon. You used a codon chart probably last year. That's where you took and looked for like the first letter, then the second letter, third letter. They came together and created an amino acid. So if you had the sequence like AUG, you would look at a codon chart and you would say, okay, what does AUG make? Well, the AUG is found on the mRNA sequence. So with my mRNA down here, if it reads AUG on my bases, the anticodon, which is located on the tRNA, would have the opposite. That's why they call it anti. It would have the opposite bases. So the A, just like it always does, would be a U here. The U, there would be an A, and then the G, there would be a C. And so the anticodon is what brings the amino acid into the ribosome, so we can keep adding amino acids onto the chain. But what the anticodon is going to do, it's going to match with the codon that's there. And then that anticodon is actually going to have specific amino acids attached to it. And so the amino acids that is attached to it would be the amino acid that would be AUG, and that would be MET or the start codon, which we'll talk about in just a minute. So methionine is going to be the amino acid that would correspond to that. Okay. The process of translation happens on the ribosome. You can see it over there on the right-hand side. A ribosome is made of 2 subunits. Okay, so there's 2 parts to a ribosome. The top part is just called the large subunit, and then the bottom one is called the small subunit, made of two parts that are going to come together when it comes into contact with the mRNA strand, okay, that's developing. And we talked about in transcription how at the end of making the mRNA, we saw pre-mRNA, we had to add that 5' cap in the poly A tail. Well, those two things are going to help the ribosome to recognize the mRNA strand for those two subunits then to come together to interact. And you'll notice something else on these ribosomes, and I don't know if you talked about this last year or not. You may have. But there's an A site, a P site, and an E site. Okay, I know that seems weird because it looks like you would read it left to right, EPA, but we actually read it APE. Because what's going to be happening is that this is going to be our strand that is going to come in here. As our strand works its way through, we are going to add new, it's going to be going, as you can see, in this direction. It shows you that little arrow right there. As the strand comes through the ribosome, what we're going to see is that the A site is where our amino acid first attaches, and I'll explain this in a little bit. And then it is going to interact with the P site. It's going to add on an amino acid. And then eventually into the E site, after the amino acid's been added on, then the tRNA that no longer has an amino acid will leave in the E site. So it actually goes from A to P to E. So the A holds the amino acid. So the A site holds amino acid. I'm just going to put AA for amino acid. The P site is going to hold the polypeptide chain, so I'll just put poly chain. So the polypeptide that we're making, remember, many amino acids make a polypeptide. And the E site, properly named the E site, is where it's going to exit. Okay, so the exit for the tRNA, it's no longer needed. We already got the amino acid we needed, so it's going to leave. Okay. So just as with transcription, translation is going to have 3 steps as well, and they're the same as our steps of transcription, but different things are going to take place with each step. The first step is going to be initiation. Initiation is going to be where the small unit binds to the mRNA, and then the tRNA that's carrying the proper amino acid for the start codon is going to come in and attach to our A site. And then we are going to see the large subunit then come in and attach as well. Okay, so again, first thing is small subunit binds to mRNA. All right. You can kind of see that up here. You can see that that small unit on the bottom is going to attach to the red strand of mRNA. The second thing that's going to happen in initiation is that the tRNA carrying the start codon, which is MET, that's our start codon, methionine, is going to attach to the A site. Okay, so you can see here is the A site. Let's assume that this is methionine up here in this diagram. If it's carrying the methionine, then it's going to come here to the A site and attach, and then it will eventually be transferred, and we'll talk about that in the elongation stage. Okay, and then the third thing that's going to happen, if we have the small subunit and we have that tRNA, the large subunit then will attach after we get the tRNA. So it's going to come along, attach as well, and that is going to initiate the process of translation. Okay. All right, the second stage of this process is going to be elongation. So elongation. And an elongation is really what you're seeing on the diagram here at the right. Okay, so what's going to happen is that we are going to have that mRNA strand, And I'm just going to show it very, very vaguely as an mRNA strand. And as that mRNA strand is going to be read, you're going to have our small subunit down here, the larger subunit attached up here. Okay, this mRNA strand at the A site, we are going to see a tRNA coming in. So let's assume that elongation, we already have a transfer RNA at the P site, okay? And so I'm going to kind of color it in here. I'm going to call the P1 here. The P site is going to be in blue. At the A site, we are going to have a new, okay, tRNA, and that's what these are. These are tRNAs coming in. And so as that strand is going through here, and remember the mRNA strand is going this direction, as the strand is being read, okay, each time we get to a new piece of that strand back here, okay, because we're going to keep getting to a new piece as it runs through, each time we get to a new piece, we're going to have to read those 3 bases on the mRNA strand to know what amino acid we need next. And so attaching to this A subunit of our tRNA, we're going to have some amino acid here. So I'm just going to kind of show it. Yes, that's very big, I know, but again, my drawings aren't great. And on the P site consists of the amino acid chain that's already been growing. So we already have amino acids attached here in the P site. The E site usually remains empty because remember, once we're getting to that E site, we keep moving these tRNAs down. Once we get to that E site, we are basically going to see an empty slot. It may still be there bound, okay? But for the most part, the polypeptide chain is stuck to the P site. The A site, we brought in the amino acid. And so this E site, the tRNA, is going to detach from the mRNA strand, and it's going to kind of be gone. Okay? So again, if we start out with our A site being filled by a new amino acid, tRNA, what's going to happen here is as we move on through this process and that RNA strands moves through the ribosome, we're going to get to the point where if this is our small unit, this is our large unit on our ribosome. Eventually, that tRNA that was in the A site is now going to move to the P site. And when it does move to the P site, it is going to then add this amino acid onto the existing chain as it moves to the P site. So our amino acid chain is going to begin to grow now on this tRNA, which I don't know why that just erased. Okay, let me redraw that. My drawings are so lovely anyway. And so now what's going to happen is the amino acid that it had attached to it is now going to attach to the other amino acids that were there already from the P site. The one that is now at the E site is going to be the tRNA that was originally had the chain. Okay, so it's actually moved down a spot and it is getting ready to exit this process. All the while, from our mRNA strand that we had that was growing, remember that mRNA strand that's through here and it's going this direction. As our mRNA strand is moving, so are our tRNAs with it. The next thing that's going to happen, as you can guess, is going to be that another tRNA is going to come in and take a spot in that A site. It is going to bring with it an amino acid, whichever amino acid it is supposed to bring in according to the messenger RNA that is with it. It is going to bring with it its own amino acid. Okay, and then this process will continue. And then this at the A site, it'll join its amino acid onto the growing chain. It will move over one. The one that's at the P site now will move to the E site and exit. And then these things will be reused over and over again, okay, until this process is over. And this is how elongation occurs. And so you can see it up here even. This is a little bit nicer drawing than mine is. You can see that we have our tRNA come into the A site. It transfers its amino acid to the growing chain so that this A site tRNA moves to the P site, which is what's kind of showing you down here, okay, is going to happen. It's going to attach to the long chain. It's going to move over to the P site, and the one that was originally in the P site is going to move and exit. And then a new one will come in and take the place in the A site. That's elongation. How do we know when this process ends? Well, it ends whenever it reaches what we call a stop codon. And so hopefully you went over last year, there are start codons. That's how we know when to start reading. And there's stop codons, and that's when we know how to stop readings. And so the third and final part of this process is called termination. Okay. And termination is going to be reached when we get to a stop codon. Stop codon. And it's going to be reached at the A site. Okay. And so if we have our ribosome and we have our mRNA, and in our mRNA we have reached here the stop codon. Okay. What is going to happen is that the A site, we are going to see our A site tRNA is going to carry with it an amino acid. Okay. Once it reaches that stop codon, however, it is then going to no longer, that is going to be the end of the growing chain. Okay. It is no longer going to add any more of the growing amino acids to the polypeptide chain. And so I'll show you over here, we have our amino acid chain. The stop codon is going to be transferred. And then once it is transferred, remember we move to the P site. We have our polypeptide chain that is going to continue to exist. I know that's a bad drawing, but notice nothing is going to come here to the A site. And what's going to happen now is that this amino acid chain, this polypeptide, will detach completely from the tRNA, and it will become a free-floating chain of amino acids that will then be coiled and folded into a polypeptide. So we'll have an amino acid chain, breaks off from our tRNA at this point once the stop codon is reached, and then the two subunits of the ribosomes will also dissociate with each other. Okay. Now, once the two ribosome subunits are dissociated, no big deal. They will be recycled. Okay, they don't get destroyed. The nucleotides that were read on our mRNA, okay, they will not be destroyed either. Okay, they'll be broken apart and then they'll go back into the nucleus to be reused for another copy of mRNA in transcription. And so all of this stuff is recycled. Imagine if we had to make this every time this process occurred, it would be a lengthy process. And so it's nice that we can recycle all these pieces and parts every time that this is needed. The other thing that I want to mention about this process of translation, a single RNA strand, an mRNA strand, can actually be read by multiple ribosomes at once. And so it's not just like we see here, here's the strand and it's going to go through the ribosome and the ribosome is going to read it as it goes through. It's going to be a little bit different. What we're actually going to see in this process is this is my mRNA strand that I need to have read. We're actually going to see multiple ribosomes reading this at once. And so each ribosome, remember, is making its own polypeptide chain. And so out of these ribosomes, not only were we making one protein from one strand of RNA, we can make several proteins from one mRNA strand. And so we can read this mRNA strand multiple times in each ribosome, and we can make not just one, but multiple proteins. Sometimes we can make up to 1,000 proteins from one mRNA strand. And so please make sure that you understand that this is not just one ribosome for one mRNA strand. We can have multiple ribosomes for one mRNA strand that just keeps reading and making proteins, which is why it's kind of a good system. It allows us to make a lot of proteins and not have to make a new mRNA strand every time we need a new protein. The only other thing I wanted to tell you guys was to make sure you take a look at slide number 63 for your chapter 17 to look at the differences for this between prokaryotes and eukaryotes. There are some slight differences between transcription and translation. So again, just take a look at that before you start doing your work. If you have any questions, as always, email me, get on the Teams app, ask me. But hopefully after transcription translation that we just talked about, this will make a little bit more sense. All right, guys, see you later.